Yes, I mean, I was, as the person who doesn't research in dementia, at least not yet, asked to give a few thoughts on lessons from both the recovery trial, but also from some of our previous cardiovascular trials. Next slide, please, Paul. So to start with the truism, we need good evidence when we're making treatment decisions. Trying to make decisions when we don't have good evidence, either because trials weren't done or they were done badly, actually can be harmful. And that may be true at the clinical coalface, but it's also true earlier in drug development. Next slide, please. And so here's my sort of, if you like, clinical trialist's view of where we are with neurodegeneration and dementia. First of all, that it's a common problem, it's a big problem, but there may be multiple rarer subtypes. Second, that it tends to be of longish or very long term with variable courses. Thirdly, that it's multifactorial, so there's likely to be a few common causes and many rarer causes. And finally, that there are multiple manifestations, whether it's about symptoms, function, quality of life, clinical, and of course, ultimately death. And that means to me that although there may be some treatments that have a big effect on a rare cause, number one, they're going to be quite challenging to find. And number two, their impact overall on a population scale problem will be pretty small. Second is that a modest effect on a common cause would be profoundly important. But of course, many of the treatments we suggest will have no impact at all overall, or in particular subgroups, or on particular consequences. Next slide, please. And so differentiating modest and important effects from no effect at all really does require randomization, and it will require large numbers of relevant outcomes to minimize the play of chance. And that's really means that we have to think about enrolling a large, often diverse population, thinking about making sure that the patients we do randomize adhere to their therapy, that we get complete follow-up, that we get unbiased assessment of outcomes and intention to treat analyses. And in a sense, those are all the basic statistical principles that underpin randomized trials. So if we've known this for a long time, surely we've solved this problem. Next slide. And what we can see is that the high cost of trials is really driving industry focus away from treatments of major causes of morbidity and mortality. So here is data from 2000 and 2015 in the US, the top 10 drugs. Revenue increased 2.5-fold. The number of people for whom those drugs would be relevant reduced about 7.5-fold. So there's nearly a 20-fold increase, if you like, in the cost per patient. And if you look at the drugs at the bottom, and I deal with generic names, not brand names, but if you look at these names, essentially this is shifting away from drugs from common disease to rarer diseases and subtypes of disease. Well, why might that be? Well, there's a number of possible reasons. Regulatory might be some of them. Maybe the trials are just becoming more complex, or maybe actually that there is a system which is motivated to both overinterpret the regulations and to drive complexity. Next slide, please. And here's the evidence for such a challenge. So the contract research organization market, since the creation of the GCP guidelines back in the early 1990s, has risen, the value of that market has risen exponentially and seems to be growing at about 10% per year from here on. So these numbers are extraordinary. And if the motivation or the business model is time and materials plus a margin, the motivation to simplify and streamline and focus is essentially non-existent. And that means that our trials are getting smaller and smaller, and our ability to understand and evaluate treatments is getting more and more restricted. Next slide, please. So Rory Collins, Louise Bowman, Richard Peter and I wrote this paper in February 2020, 6 weeks before in fact, actually 4 weeks before I got diverted by COVID, when we talked about the magic of randomization versus the myth of real world evidence. Now, I think if I had my time again, I'd slightly rephrase that and I'd say the magic of randomization when one does randomization with real world data and within the real world, not just the data, but in real world clinical settings. The summary of the paper is on the next slide, please. Essentially, what we argued was that if one gets smart design and smart design of the delivery of a trial, is able to integrate with routine healthcare and data systems, and proportionate regulations and guidance, and indeed application and interpretation and inspection of those guidelines, then one can have a substantial improvement on better patient care and public health. Now, on the next slide, I just wanted to sort of summarize some of our previous work on cardiovascular trials and sort of make a few points that I hope might be relevant to today's discussion. This first trial is a trial of lipid lowering in chronic kidney disease. Chronic kidney disease is quite interesting to think about. Most of it is a collection of relatively rare or very rare diseases, and then there are some big causes like diabetes too. But what this shows is that addressing a common pathway, in other words, lowering blood cholesterol, can reduce the substantial risk of heart disease in these patients. So just because it's a cluster of rare disease does not mean that there isn't a treatment that might actually affect a common pathway and have an important impact. Next one. Another example from kidney disease, but this time kidney transplantation. And this is really illustrating the fact that actually one can get, on this slide, 6 years of follow-up, and indeed on to 10 years of follow-up, as we're at about now, to look at the impact of a short-term intervention on transplant survival. We were also able to look at safety, at cancer, infection, on other causes of mortality, and see this clear separation between the two lines. So again, good trial design can get you to understanding what are the long-term impacts are of short-term therapies. The third point. Here is a trial of CETP inhibition, another lipid modifying drug in heart disease, making the point that some disease processes take time to, or modifying some risk factors can take time to modify disease outcomes. So here you can see that this trial of lipid modification, basically lowering cholesterol, it took 2 years before the curves started to separate. Why? Because the heart attack you have tomorrow is driven by the behaviors and your cholesterol levels that you had yesterday and years before. And it takes time for the disease to be modified. So let's be careful not to make our trials too short and miss out on benefits. In fact, actually on this example, another company did exactly that. They stopped their trial at 2 years for futility and missed a big opportunity. And finally, on this slide, not all drugs are safe, and not all drugs are safe even when you think they're safe. So here is a trial of 25,000 people randomized to niacin. It's a B vitamin. It was on the back of your box of cornflakes this morning, but it's given in gram doses to modify cholesterol levels, and has been licensed for the best part of 50 years. And what we found in this trial was, one, it doesn't lower the risk of heart disease, and two, it does increase the risk of major bleeding and major infection, the sort that requires people to go into hospital. That had simply not been known in 50 years of routine pharmacovigilance, observational studies, and clinical use. So randomized trials can tell you things about safety that you weren't expecting to know too. Next slide, please. And how can one deliver these trials? Well, data and the smart use of technology is certainly an important element. So here is an illustration. The Orion 4 trial is a trial of people who've already had an MI or a stroke, giving them twice yearly cholesterol lowering injections versus placebo. We were able to work with NHS Digital, find where the biggest sites were by numbers of patients across England and Wales. We've done it in Scotland subsequently. And in fact, then we were able to invite precisely those people to join the study. Next slide, please. Secondly, central statistical monitoring. One can use the data that one's collecting in order to identify outliers. And this is a sort of I'm sure there's a better name for it, but if you think of it a bit like a Manhattan plot, on the y-axis is the minus log p-value. The blob size is proportional to the number of patients at a particular site, and each blob is a site. And what you can see is that many of the sites in yellow at the bottom are performing just average, and there's no particular concern. But there are one or two sites up in that top right-hand corner where they're really markedly different in what they're doing from the others. Now, if you have a limited number of monitors and a limited amount of budget, and those two statements are always true, then you would send them to those top right-hand corner sites rather than the massive yellow. You could substantially improve quality and reduce the cost. Next bit, please. Third example, this is a Scottish study of diabetic retinopathy, so moderately common but not wildly common. And here are over 1,000 patients randomized and recruited using hospital or diabetes records in Scotland and invited from their homes. And the map of where these people are is the map of where people live with diabetes. Next slide, please. And then finally, you can use routine data to follow patients up. Here's a trial of people with diabetes who are treated with aspirin versus placebo, or fish oils versus placebo. The blobs in black and the results in black are the full trial results, carefully adjudicated, etc., etc. And the ones in grey are simply, well, what would happen if we used the routine hospital discharge data and did no adjudication? The answer is you get the same answer for a fraction of the money. Next slide, please. So that's our cardiovascular story. What about the COVID story? Well, it's remarkably familiar, of course, but here's a major public health crisis which kills 1 in 4 patients who come into hospital. There's huge uncertainty about which treatments might work. There's unlikely to be a single big win, and large-scale randomization would be the smartest way to find effective treatments. Next slide. Now, this is an extreme example, if you like, of how simple a trial can be and how compelling the results can be. But it just, in a sense, by showing the extreme, shows what can be done. The randomized trials don't have to be complicated, but they must be practical. As you can see, simple randomization. The eligibility was patients admitted to hospital with COVID. We focused on an outcome that mattered, in this case, death. One-page case report form, extensive linkage to routine NHS data to ensure complete follow-up. Next slide, please. Over time, we were able to modify the randomization structure actually to include multiple factorial comparisons, so we can study multiple drugs at once and drive the efficiency even further. Next slide, please. Now, COVID can affect anyone, so recovery was open to everyone, regardless of geography, as shown on the left, and regardless of age or ethnicity, comorbidity, and so on. One of the things I want to illustrate from the slide on the left is that although there were 1 or 2 enormous centres, the biggest blob in the middle is Leicester, who recruited about 1,500 people in that one centre. Actually, there's a substantial recruitment coming from district general hospitals, and that actually drives diversity in the types of patients who are enrolled. And on the right, recruitment does not have to be slow. Now, of course, with a pandemic, the virus or the pandemic ebbs and flows like the tide, and you can see when the tide is out, there was not much recruitment, but when the tide's in, recruitment can be very rapid indeed. Next slide. And to illustrate the point about linkage through to routine healthcare data, here are 25 different data sets from across the UK that are incorporated to assess everything from whether patients have been discharged from hospital, whether they're on a ventilator, whether they're alive or dead, what their background medical history is, what their COVID testing status is, and so on. Next slide, please. And so to the results. I mean, the first thing I want to illustrate, I mean, this result's quite well known, I guess, now, that dexamethasone, a cheap and available steroid, reduces the risk of death by 1/3 for people on a ventilator, by 1/5 for people on oxygen, and not at all for people who are not on oxygen. Now, a couple of things there. Number one, people told us don't randomize to dexamethasone, it's dangerous to give steroids. They also told the MHRA that, and it took substantial effort to make sure we could keep this trial going. So not all hypotheses, they may have good, if you like, evidence behind them, but they then turn out not to be supported by the evidence from the trial. But I think what we see here is a combination of clarity and precision. Number one, these are very clear results. We know what the answer is in each of these types of patients. And number two, we weren't expecting necessarily that we would see such strong heterogeneity between the different types of patients. But it was the data, is by enrolling very large, broad range of people into the trial that we were able to find that both the whether there are benefits at all, and secondly, how big those benefits were in proportional terms, let alone absolute terms, varied by the patient type. Next slide, please. We've done that repeatedly. And so, for example, if you look at Ronapreve down the bottom right-hand corner, this is an antibody combination drug given to people in hospital in this context, in this trial. And one of the things we see is that if you gave antibodies to people who have not got antibodies, that's the sort of top two lines on that bottom right-hand corner, then there's a substantial reduction in their risk of death. People have told us that the virus was sort of history, and dealing with the virus was history by the time people were in hospital. Actually, that turns out not to be true. The second thing you can see is that giving antibodies to people who've got antibodies is expensive and frankly useless. So again, what one's seeing is this combination of clarity and precision. Next slide, please. And just importantly, are all the good hypotheses that actually turn out to be unfounded. Famous drugs like hydroxychloroquine, many advocates for that, not all of them political, some scientific. Convalescent plasma given to 100,000 Americans in a large, simple observational study, because apparently it was too hard to randomize. Again, turns out to be, to all intents and purposes, useless in this context. So not everything we study works. In fact, probably more things that we study don't work than do. But actually, this is a very robust way of testing hypotheses. Next slide. And so overall, these are the numbers of patients in the active arm for each of these comparisons, including, I'd highlight for today's discussion, what you might call it as essentially a phase 2 trial of 360 people randomized to dimethyl fumarate versus 360 randomized to usual care. So it is possible to do smaller mechanistic studies. The outcome in that study was one around recovery time and oxygenation. Next slide, please. So how is this viewed, and how is this viewed from abroad? So here is Zeke Emanuel writing in the New York Times and reflecting on how the UK managed to do this and others didn't, particularly the US. Number one, it was designed to be easy to take part in. The second, it was quickly approved and adopted. The third, that we had data. The fourth, that actually we had leadership. The fifth, we had research nurses. And the sixth, that we incorporated this in part of everyday clinical care. Now, not all of those elements will be true, necessary, or relevant for all contexts, but they're very useful to keep in mind as one's thinking about how could we change the landscape in dementia. Next slide, please. So not all trials have been successful during COVID. In fact, here's an analysis from Janet Woodcock, who at the time was interim commissioner of the FDA, now deputy commissioner. She reviewed all the trials registered on clinicaltrials.gov and the WHO Clinical Trial Registry, 2,800 of them, and concluded that only 5% were randomized and adequately powered to answer their question. So in other words, 95% of the trials, give or take, were designed to fail. And so I think that what we need is not more trials, but a few more trials which will actually provide meaningful answers. And I should say, when I'm talking about meaningful answers, I don't mind whether that's a meaningful answer about clinical licensing or a meaningful answer about how one might further develop the therapy or the therapeutic area. But first ask a good question, and secondly, get the right answer. Next slide, please. And that's been taken up. I mean, it was taken up by the G7 when UK had the chairmanship last year. So I think it was very early June last year. There's a meeting in Oxford of the health ministers, recognizing clinical trials are the primary way to generate actionable evidence, recognizing on the basis of that Janet Woodcock paper that many trials were inadequate in size, design, and conduct, failed to generate evidence needed, and making commitments to move forward and improve the situation. Next slide, please. Patrick Vallance produced a 100 days mission to respond to future pandemic threats, which I actually think is a very strong analysis of what's needed in a number of areas, including areas around drug discovery, around manufacturing, around data and trials, which I was involved in, and also around finance, frankly. Next slide, please. And in that was what was an area on clinical trials, which I helped co-write, but it included this commitment. We should refocus regulatory guidelines on the fundamental scientific and ethical principles that underpin randomized trials, whilst embracing flexibility and innovation. GCP should be revised to focus on what matters rather than what is easy to check, and place an emphasis on principles and purpose rather than process. I argued at the time that this wasn't just for pandemics, this was for all time. And if you look at the next slide, please. I'm pleased to say that the World Health Organization at the World Health Assembly last week, if you essentially double down on this, and pretty much the exact wording that I have just read out to you is now included in this paper, which has been adopted by the WHA last week. Next slide, please. For my own work, I've long been interested in the ability of good regulation and good clinical trial guidelines to guide, and bad regulation and in particular, a bad interpretation of regulation and guidance to substantially impede our ability to make progress, whether that's around many of the, if you like, common and ordinary things around clinical trials, or whether that's as we look ahead around the use of data and the use of technology. And one of the problems with most regulations is that they're written by a small group, typically of one subset of the regulators. So one subset might be, the GCP rules get written by the inspectors. It's a bit like, the police writing the laws, interpreting the laws, possibly over-interpreting the laws, and then indeed in due course being judge, jury, and proclaiming the sentence. It's not a very healthy way of doing things. It's not it doesn't engender trust. And it also means that one doesn't take the full range of opportunities as the world is moving forwards. And that's true whether one's talking about data and technology. It's also true in medical advances, and particularly true as one's thinking about the different relationships between patients, members of the public, who might not consider themselves patients, at least not yet, and industry, academia, and indeed those who are charged with regulating. So this is not a criticism of regulators per se, but really what it seemed important to me to do was to make sure that we actually produced and co-developed guidelines that really were fit for purpose and did focus on those principles. Next slide, please. And so I'm not going to go into the detail now, but essentially these 5 principles have been published online on our website, which I think was on the previous page. Number 1, get the answer right. That's why you do trials. If your trial doesn't answer the question, then the rest is, in a sense, immaterial. Number 2, of course, look after the patients. Number 3 is around collaboration and transparency. Number 4 is around feasibility. And number 5 is ensuring that the quality is managed effectively but efficiently. And as I say, these are now online. They have been, they do feed into that WHA paper that we've just looked at. And I think many organisations are considering how best to take these forwards. Next slide, please. And so lessons for the future. Well, these are slightly COVID lessons, but I think they're true. Arbitrary use of unproven treatments is damaging. Randomized trials are a critical component of high quality clinical care. And I think we can think about that in the context of dementia. And compelling results change practice. But we've got to design our trials so that they will deliver actionable results, actionable about whether to develop a drug further, actionable about whether to use a drug, actionable and whether to license a drug, and actionable in terms of how much one should pay for it. They need to be feasible, inclusive, and focus on what matters. Next slide, please. And it's really for those reasons that we set up Protest as a not-for-profit organisation, thinking about these elements back to our New England Journal paper about smart design, thinking about the efficient use of data and technology, integration of routine data into the randomization process, software engineering to drive clinical, scientific and operational efficiency and quality, central statistical monitoring, I showed you some of that, and adaptable and extensible to different circumstances and different needs now and in the future, but also effective partnerships with patients, clinicians in the healthcare system, with pharma and other medical industry, and finally with regulators, payer assessors and purchasers' perspectives. So I'll finish there on my last slide just to say thank you, and I'm happy to take any questions.
Thanks so much, Martin. That's wonderfully provocative as well, especially about the sort of proportionality of trial regulations. We've got a few questions in the chat, but maybe I could just quickly open with one. Obviously, recovery was unusual in the sense that outcomes could be measured relatively rapidly. And in the dementia space, that's one of our problems, is outcomes have often taken a long time to measure. Have you got any suggestions or advice for that in terms of how we could improve that?
Yes, I mean, I think, you know, different trials are different, different diseases are different, but as I showed you in some of that cardiovascular work, I mean, most of the trials I've ever run have had 5-year outcomes, and then we followed on for 10 years and plus. When one's thinking about that, what are the key lessons? Number one is, if it's a chronic therapy, how do you keep administering the therapy? Number two is ensuring no loss to follow-up, in particular, no loss from one group versus another, but really ensuring loss to follow-up. And number three is thinking about what are the outcomes that will matter in 5 or 10 years' time. And they're not necessarily the ones that matter in the short term. So in the short term, to go back to my cardiovascular examples, one might be interested in, can one reduce the risk of MI and coronary revascularization? In the longer term, one's interested in, can one reduce the risk of cardiovascular death? I mean, of course, if you could see that early on, that would be great. But the reality is that, you know, cardiovascular death is usually the last thing that happens to people. You know, non-fatal MI and so on will be a little earlier. So one may get signals earlier, but even if one gets signals, even when one hits a primary outcome, you still want to know about the long-term efficacy, and you also want to know about the long-term safety.
Okay, I've got another question here. This one's from Vanessa Raymond. She says, given the likely need for good linkage with long-term NHS datasets in platform trials, how can we marry that with the current lack of follow-up that normally occurs for patients with dementia in primary and secondary care? Is there a way in which the infrastructure can be improved?
Well, I can't comment on how patients are followed up in routine clinical care and whether that's appropriate or not. I think that's for the clinicians here to think about. But you know, there is a lot of data on what happens to people. I mean, you know, the NHS does collect data on people essentially throughout their life. There are still some challenges about where those data are. You know, NHS is one logo, essentially one funder. And after that, everything else is a complex mess of multiple organizations overlapping, each with their own governance. I mean, that's the reality of the NHS. But actually, you know, if we could get hold of GP data for these purposes, and in COVID we could, if one can link through to the hospitalization data, and obviously one can do to the civil registration, to the mortality data, there's a lot one can get. One thing I would say is that you may not get the perfectly detailed information you need from the routine data. But the key challenge in long-term follow-up, and particularly for large-scale trials, is knowing the difference between 1, something happens to this patient, 2, nothing happens to this patient, and 3, I have no idea whether anything happens to this patient. And you really want to absolutely minimize the third one. And so if you can get an indication that a patient was admitted to hospital with something that sounded, you know, relevant to a dementia outcome, then you can go to that hospital and dig through all their notes and pick out their scans and do all the things that you would want to do to get the detail. But you can do that because you know that it's like water divining. You know there's water down there. And so, you know, that's the you can layer things on, but if you had to go looking through all the CT scans and the MRIs and whatever else in every patient all the time, that's an infeasible task.
Yeah. Okay. Time is pressing, Martin, but I'm just going to give one more question. This is from Rory Collins saying, could you comment on the issues with regulatory auditors not focusing on what matters?
Yes, I mean, I think there's an issue around training and experience, but I think there's a real issue about what matters to the interpretation of the results. And I think that in particular, of course, lots of detailed records can be checked, but think about the routine healthcare data. In recovery, we've got, I think it's 6 or 7 different sources of information about whether someone's alive or dead. They're not all saying the same thing at any one time, and actually, there's a good reason for that. Wife may tell you that my husband died. There may be a coroner's inquest, and the death certificate may not get issued for 6 months. You can bring those data together in a way that gives you very robust information about the impact of a treatment on death. But you shouldn't expect each record to be identical to the other, and you certainly shouldn't expect an investigator, for example, to change their case report form because some other data source says something different. I mean, that's like going to an eyewitness of a car crash and saying, change your statement. That's not a sound route to go down.
Okay, thank you very much, Martin. I'm sure there are many other questions, but in the interest of time, we have to move on. But thanks so much for joining us and sharing your experience with us today. Thank you.